Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at Core Practical 3, which investigates membrane structure, including the effect of alcohol concentration or temperature on membrane permeability. So, cell membrane permeability is affected by temperature and pH. The following practical will be using a beetroot to investigate the effect of temperature and pH on cell permeability. Beetroot cells contain a dark purple-red pigment called betalain, and the leaking of the pigment increases with an increase in the permeability of the beetroot cell. A colorimeter can be used to measure the amount of pigment in a sample. A colorimeter is a machine that passes light through a colored liquid sample. It then measures the amount of light absorbed by the sample. In this case, the increase in absorbance indicates a darker solution, which suggests an increased permeability of the cell membrane. So the colorimeter must be zeroed before each use. This can be set to using distilled water. And this is how you calibrate the colorimeter to ensure that all your results are the most accurate it can be. So what do you need for this practical? You're going to need a scalpel, cork borer, cutting board, ruler, digital balance, test tubes, measuring cylinder, water baths, digital stopwatch, pipettes, and of course colorimeter and the cuvettes. So using a cork borer and a scalpel, cut the beetroot into equally sized sections. This will act as the control variable as the surface areas and volumes can affect the permeability rates. The cork borer ensures that the diameters of the pieces are the same, so that acts as the control variable, and a ruler can be used to cut them into the same length, once again ensuring the control variable. Add the beetroot pieces to 5 different test tubes, each containing the same volume of water. Once again, another control variable. However, the temperature in each of these different water baths should be different. So this one, the temperature, is the independent variable in this practical. And set the timer for 30 minutes. Remove the beetroot pieces when the time is up. Use pipettes to transfer the pigments to the cuvette, one at a time, to measure the absorbance. Ensure that different pipettes are used each time to avoid transfer of pigment. So, looking at the results, so a graph can be plotted from the results with the different temperatures on the x-axis and the absorbance on the y-axis. What you will notice with your graph is that as the temperature increases, the membrane permeability also increases. This is because as temperature increases, the phospholipids within the cell membrane move more as a result of higher kinetic energy. The phospholipids are not as tightly packed, and this results in an increase in permeability. At high temperatures, the membrane proteins also denature, further adding to the increase in permeability. However, you might notice an increase in permeability even in low temperatures, and this is most likely because of ice crystals that form, as these crystals can paste the cell membrane, hence adding to the increase in permeability. And this is something for you to look out for when you do your practicals. So what are the limitations of this practical? So make sure that you use the same cuvette for every reading. Different cuvettes can have different thicknesses, and that can affect the results. Your results might be affected if the cuvettes are scratched as well, so that's also another thing for you to consider. There is also the possibility of the size of the beetroots not being too accurate. So once again, affecting the permeability, and different parts of beetroot tissue can have more or less pigment, hence the results obtained can be affected. The environment can be altered such that you can look at the impact of different factors on the permeability of cell membranes. So you can, for example, look at the effect of different pH levels, or you can look at the impact of alcohol. Except you need to change your experiment in little ways. So instead of looking at the changing the temperature of the water baths, what you can do is change the concentration of alcohol in the different beakers. So that's going to show you the impact of alcohol, impact of different concentrations of alcohol on the permeability of the cell membrane. Or if you want to look at the pH levels, you can do the same, but once again, you, you can vary the pH levels in the different solutions instead of the temperature. And what you will notice is that the trend will be very similar. As alcohol concentration increases, the permeability of the membrane will also increase. And this is the result of alcohols dissolving in the lipid and cell surface membrane, and the cell membrane loses its structure and the beetroot pigment leaks out. Professor Snap today. Hope you found this topic useful. If any questions, please let me know in the comments below. And don't forget to like, share, and subscribe.